Hello, this is Andrés Martinez from Argentina, and together with Maité, Bárbara and Juan, would like to show you some of our results on the effects of relatedness and density on the formation of yellow-jacket Vespula germanica male aggregations in Patagonia. Vespula germanica is a social wasp that represents a problem in many parts of the globe. Originally from Eurasia, it has invaded many parts of the world, including Argentina. The wasp was first detected in 1980, and since then it has spread aggressively throughout the country. The species has a complex social behavior, with mated queens going through winter in solitary form, and in spring they start the new nests with the help of workers. The colonies grow until autumn, and before they disintegrate, the new queens and drones leave the nests to mate. When this happens, drones form aggregations to mate with reproductive females. Since the reproductive period is very short and chances of encountering related mating partners can be high, mechanisms that help minimize inbreeding can be important for the success of yellow-jacket populations. This is because inbreeding in Hymenopterans can reduce the fitness of the progeny, since their single-locus complementary sex determination system may generate diploid males and result in infertile triploid progeny, representing a cost for the colony. Our objective was to establish the effects of relatedness and density in Vespula germanica drone aggregation behavior. We hypothesized that genetically diverse male aggregations could help minimize the negative effects of inbreeding, and predicted that drones would avoid high densities of nestmates while not avoiding non-nestmates. We conducted our experiments in an olfactometer, where we assessed the response of drones toward different olfactory stimuli. A single drone was placed in the central arena of the olfactometer, where we recorded its behavior toward different treatments presented inside the odor chambers. We tracked the movement of the experimental drone in the central arena of the olfactometer and calculated the proportion of time spent in each of the odor areas. Each of the squares here represents the path made by one drone, where it was possible to calculate the time spent within each odor, represented here as red or blue. We did a total of 9 assays, each one with 32 experimental drones. As stimuli, we used clean air or different numbers of live drones, either nestmates or non-nestmates, in equal numbers of 2, 6, or 10 individuals. So, for example, in assay number 9, we tracked the response of one drone toward 10 nestmates versus 10 non-nestmates. Here we see the results of the 9 assays in terms of the proportion of time spent in each of the different combination of odors and the different densities of nestmates and non-nestmates. Here we see, for example, the response toward 10 nestmates versus 10 non-nestmates. The time in clean air was greater than the time spent with nestmates at all densities. We recorded a similar response for air and non-nestmates at high densities, but the time spent in air versus 2 non-nestmates was not significantly different. Interestingly, when nestmates and non-nestmates were presented simultaneously, no statistical differences were detected under all densities. However, a trend to avoid nestmates was observed at the lowest density. Our results suggest that drones would avoid aggregating with their nestmates at all densities, while non-nestmates are avoided only at high densities. These two factors could be important in regulating male aggregations, resulting in the conformation of leks with a larger genetic heterogeneity. Finally, we would like to thank the different funding bodies that contributed to this study. Thank you for your time.